We're going to look at the different processes that occur before gastrulation during mammalian development. So, in early mammalian development, the oocyte moves from the ovary to the fimbriae to the oviduct. So, if we look at the figure, we can see the ovary and we can see an egg or the oocyte being released from the ovary during the process of ovulation, and it through the fimbriae it is able to go to the oviduct. Now, fertilization occurs in the ampulla region of the oviduct, which is shown in the blue box. Once fertilization occurs, a microRNA that is made in the sperm is able to initiate S phase in the zygote. Now, the oviduct have cilia that move the embryo to the uterus, and during this process, the embryo is actually undergoing its development. So, the zygote starts the cleavage step as the embryo moves towards the uterus. And that is what is shown here, that the embryo undergoes cleavage while moving to the uterus. Now, how does cleavage occur in mammalians? So, cleavage is a holoblastic rotational cleavage. The first cleavage is a normal meridional cleavage. However, in the second cleavage, one of the two blastomeres divides meridionally and the other divides equatorially. And we actually saw this type of cleavage when we learned about the cleavage in C. elegans, the nematode worm. So here in this picture, we are seeing how rotational cleavage is occurring. So we can see that the two cleavage planes are actually perpendicular to each other, where in one case, one of the blastomere divides meridionally, while the other divides equatorially. Now, in the case of mammals, the cells show asynchronous divisions, which means the cells don't divide at the same time. We normally observe synchronous divisions in a lot of other organisms. Zygotic transcription is activated quite early in mammals, so usually by around the third cell division, zygotic transcription is activated, whereas in other examples, we actually saw it was activated somewhere around the 10th or 12th cell division. Now, there's a lot of modification that happens in the chromatin where the histones are replaced and the DNA is demethylated in many regions. So it is mostly demethylated and sometimes even called as hypomethylated. Now, all cells at the 16 cell stage and before are pluripotent. So after the 16 cell stage, the cells lose their pluripotency. During early development, there are changes that happen to the shape of the embryo. So after the third cell division, the embryo forms a compact ball of cells, which is called as compaction. And this is what is shown in the figure, where if we look at the part in the blue box, we can see that we have multiple cells of the embryo, and they're all spread out. However, when we look to the next panel, now they have formed a very compact ball. So this process of forming this compact structure is called as compaction. Now, E-cadherins that are expressed on the cells promote the adhesion and enable compaction to occur. The cells that are on the outside, they form a tight seal by forming tight junctions, and these cells are going to be called as the trophoblast or the trophectoderm. We had actually learned about the trophoblast or trophectoderm in chapter 5 when we learned about stem cells. The cells on the inside form gap junctions, and they are the cells that will form the inner cell mass. Now, the trophoblast forms extra embryonic structures, while the inner cell mass forms the actual embryo. And the blastomeres in the 2 or 4 cell stage are totipotent, which means they can form extra embryonic structures, but the ICM cells are pluripotent. Now, the early blastomeres have the ability to become either the trophoblast or the inner cell mass. The expression of the protein CDX2 makes a cell become a trophoblast, while the expression of OCT4 makes a cell become the inner cell mass. Now, OCT4 represses the expression of CDX2, and CDX2 represses the expression of OCT4. Hence, if a cell is making CDX2, it is not expressing OCT4, And conversely, if a cell is expressing OCT4, it will not be expressing CDX2. This way, we can separate the trophoblast from the inner cell mass. Now, the Hippo pathway plays a role in determining CDX2 expression, and we will look at how this happens in the next slide. But before that, we can see that the inner cell mass forms all the cells of the embryo that I had told you before, and they are pluripotent. Now, the presence of OCT4 and SOX2 are the ones that provide the pluripotency for the inner cell mass. OCT4 associates with SOC2, and they're able to give rise to the expression of NANOG. And all these three together enable pluripotency in the inner cell mass. This is not something you have to remember, but it's better to know how pluripotency is there in the inner cell mass. But what should be remembered is CDX2 is associated with cells becoming trophoblasts, while OCT4 expression is associated with cells becoming the ICM, and how the Hippo pathway plays a role. So let us see how the Hippo pathway plays a role. Now, the outer cells tend to become trophoblasts due to the failure of the activation of the Hippo pathway. So, in the outer cells, the kinase Hippo is not sufficiently active. So, when we look at the part of the figure in the blue box, we can see that the outer cells are associated with cells surrounding them, but not on all sides. There will be one side where there is no cadherins that allow association with another cell, and that is the leftmost side that we can see in this figure. Due to this, there's not enough activation of the hippokinase. And because of the inactivation of hippokinase, the YAP goes to the nucleus. So YAP is a protein, and it can associate with the transcription factor TEAD4 and activate CDX2 expression. So when YAP is in the unphosphorylated state, it can enter the nucleus and interact with TEAD4, which will activate CDX2 expression. Now let us see what happens in the inner cells, and for that we will look at the part of the figure that is in the blue box. The inner cells are surrounded on all sides by cells, and hence they have a lot more cadherin interactions that are happening. Hence the inner cells activate the Hippo pathway pretty well, and hence Hippo is active. Now, when Hippo is active, it will phosphorylate YAP, and phosphorylated YAP gets degraded or remains cytoplasmic depending on the organism. Now that is what is shown here, where in the case of the inner cells, YAP gets phosphorylated and phosphorylated YAP is degraded. And because of that, TEAD4 does not get YAP and does not interact with YAP, and hence there is no activation of CDX2. And hence, in the case of the inner cell, CDX2 is not expressed. The absence of CDX2 allows OCT4 to be expressed really well, and thus the inner cell mass are able to express OCT4. Now let us look at the process of cavitation. Now the 16-cell embryo is called as a morula, and the morula does not have an internal cavity. However, as development progresses, an internal cavity is formed, and that is formed when the trophoblast cells secrete fluid inside to make a blastocoel. And this process of forming that internal cavity is called as cavitation. Now, in the figure that is shown in the blue box, we can now see the actual cavity that is present in the embryo, and this cavity is basically the blastocoel. Now, the trophoblast cells pump sodium ions into their inner space, and that is what promotes the formation of the cavity or the blastocoel. Because of a high concentration of sodium that is present in the inner space, water is drawn into the inner space through osmosis, creating a fluid-filled cavity. Thus, the blastocoel is formed, and the blastocoel then pushes the inner cell mass to one side, giving the characteristic structure that we see. And here in the figure, we can see the inner cell mass being pushed to a side because of the presence of the blastocoel or the cavity. Now, the developing embryo still has the extracellular matrix that covered the egg, which is called as the zona pellucida. The zona pellucida prevents attachment of that embryo to the oviduct. If the fertilized egg attaches to the oviduct, that is when we have ectopic pregnancies. And ectopic pregnancies can be quite threatening to the mother. And hence, the organisms do not want to have fertilized eggs attached to the oviduct and develop there. So, that is when the fertilized egg makes its way to the uterus, and the zona pellucida prevents attachment to any part of the reproductive system. However, to bind to the uterine wall, the embryo has to get rid of that zona pellucida. So the trophoblast cells secrete a protease to digest the zona pellucida, and the blastocyst then squeezes out, and this is called as hatching. And that is what we are able to see in the figure that is in the blue box, where we can see the zona pellucida and the blastocyst coming out of that zona pellucida. Once the blastocyst is out of the zona pellucida, it is able to interact with the extracellular matrix of the endometrium, which is the inner lining of the uterus. And so the extracellular matrix of the endometrium is able to catch the blastocyst through interactions. And that is what we are seeing here in the figure that's in the blue box, where the blastocyst is interacting with the endometrium that is present in the uterus. We can see that the inner cell mass is the part that is closer to the endometrium. Now, wind induces the trophoblast to secrete more proteases to digest that uterine extracellular matrix for implantation. Now, implantation is a very important step because in the case of mammals, eggs are not laid in the environment for development. The growing embryo actually develops within the mother, and hence it has to have a firm attachment with the uterus of the mother, and this happens in implantation. Most of the embryonic development in mammals occurs in the uterus. Now, the embryo attaches to the uterine wall through a process that's called as implantation, which is shown in the figure to the left. The uterine wall is rich in blood vessels that provide nourishment, and that region is called as the endometrium, which is highlighted in the figure to the right. It should be noted that, as we can see in the figure to the right, this layer of the endometrium, which is rich in blood vessels, can be quite thick so that it can provide nourishment to the growing embryo. If implantation does not occur and the embryo does not attach to the endometrium, then the embryo cannot survive and develop. So, to summarize, we learned about the cleavage process in mammals and the processes of compaction and cavitation. We also learned about the different cells that are formed during the early development process, as well as the processes of hatching and implantation.